Hello again. Today we're going to go ahead and finish talking about, we're going to first recap on mitosis, so interphase and mitosis, and then we're going to talk about what happens with those telomeres and what can actually occur if there are any kind of checkpoints that aren't done correctly, and if a cell can continue to divide, what are the consequences of that? Okay, so let's do a little recap first. Remember, before mitosis can occur, we have to go through interphase. Interphase, we have G1, which is where it grows. We have the S phase, which is DNA synthesizes, which means DNA gets replicated. And then we have G2 phase. That's where it needs to grow some more before it can go ahead and go into mitosis. Mitosis, remember PMAT. We actually have prophase, prometaphase, but there's also metaphase, anaphase, and telophase. And last but not least, cytokinesis is where everything completely separates into two identical daughter cells. So let's say right here, this is our parent cell. This is before interphase, okay? Right here is our parent cell. You can see 1, 2, 3, 4 different chromosomes in here, okay? Because there's actually a longer white, it looks like, and then a shorter white, and then this is actually a longer green, and then like a shorter green. Interphase, DNA synthesizes or replicates. That's what you can see right here. That's how we actually have sister chromatids now for each chromosome. Now they're going to line up during metaphase on the metaphase plate. Then right here we're seeing the sister chromatids getting pulled apart in anaphase. Here's telophase, where nuclear envelope is starting to reform. And then last but not least, cytokinesis, where if this was an animal cell, we'd be seeing a cleavage furrow, or if it was a plant cell, we'd be seeing a cell plate starting to form. So the parent cell starts off as a diploid, which means we have 2 sets of each chromosome. Remember, in our body cells, we have 46. So those are 2 sets of chromosomes. And we're going to end up, our daughter cells, which are down here, they end up diploid as well. Because if you can see, they're exactly identical to the parent. There's nothing different between the parent cell and this daughter or this daughter cell. They are all clones of the parent cell. So one parent cell creates 2 identical diploid daughter cells. So at the end of each chromosome, though, okay, there are actually what are called telomeres. Telomeres, like I said, it's the very, very end of the DNA strands. This actually has a non-coding sequence there. Okay, as DNA gets replicated, these get shortened and shortened every single time. So let's say a chromosome gets replicated one time, that telomere is going to get cut off a little bit shorter. The next time it's going to get cut off a little bit shorter, and so forth. These non-coding sequences at the end are non-coding because they're only there to protect the chromosome from any kind of it being degraded by any kind of enzymes or anything else. So once again, here it is, so you can actually see what's happening from one to the next. Remember, these are telomeres, which are the very, very ends of chromosomes. So one cell, let's say it goes through cell division, that telomere gets shorter. Then those 2 cells, the daughter cells, go through cell division, and that gets shorter, and so forth and so forth. And to the very, very end, there is no more non-coding sequence or telomere area left. After that happens, cell division completely stops, and the actual cell is programmed to die, which is called apoptosis. So this is showing you why each of those telomeres are getting cut off each time. You can just go ahead and click on this link and watch that video. It's very informative and very easy to follow along and understand what's happening. But basically, telomerase is a molecule that actually reverses telomere shortening that normally occurs after DNA replication. So instead of it getting shorter and shorter and shorter, we have an enzyme called telomerase, and that's going to actually help and reverse that shortening process. So what kind of cells would have some telomerase in it? Because not all cells can have telomerase to actually continually divide throughout their whole life. In adults, we only have telomerase located in our stem cells. Okay, they can actually, once again, reverse that telomere shortening that normally occurs. And they basically can divide indefinitely. Cancer cells, unfortunately, also have high levels of telomerase. So instead of cancer cells eventually dying off, they're actually going to continue to divide indefinitely as well. They're basically immortal cells because of telomerase. So remember, what should normally happen to the telomeres, every division, is it should get shorter and shorter and shorter until finally it's too short and it kills the cell, and it's called apoptosis. That's normal cell life. Cancer cells, they have telomeres, so it's actually protecting them and it's allowing the ends to elongate. So basically, it will never ever be too short to divide, and therefore apoptosis won't occur. So unfortunately, those actual cells will continue to divide indefinitely, and that's what causes tumors. Okay, growth factors. They're going to be the ones that actually stimulate a cell to divide and eventually differentiate into whatever the cell needs to become. We also have tumor suppressor genes, or what I like to consider brake pedals. Okay, they're going to stop the cell cycle if something abnormal happens. So just like the name is called, it's a tumor suppressor. It's going to suppress any kind of tumorous cells from actually dividing. So it's a brake pedal, okay? It's going to make any kind of mutated cells undergo apoptosis immediately so that way we can kill that cell because we don't want that mutated cell to divide. So, tumor-causing mutations. Number one, decreased activity or number of tumor suppressors. So if we have less and less tumor suppressors, guess what? There's not a lot of items that are going to stop that cell saying, nope, stop, break the pedal, push the pedal and brake. There's not going to be those guys to do that. So if we decrease the number of tumor suppressor genes, we're decreasing a lot of those mutated cells from getting stopped in cell division. HPV, which is a virus, that actually makes cells produce proteins that bind to tumor suppressors, okay? So just like how we learned about enzymes, if you have an inhibitor come in and block the active site, then what actually should be attaching there cannot attach. So that's what HPV does. It's going to cause proteins to bind to these tumor suppressor genes, and then that way it makes them inactive. They can't do their job. So once again, HPV, that's why HPV actually leads to cancer eventually, because there's nothing to stop any kind of mutated cells from continuing to divide. So very, very important to get vaccine for that, or at least to get tested for that. So remember at the very beginning of mitosis, we saw that actual cell cycle in that circle, and we went through the interphase, all those different phases. We went through G1, the S, and the G2 phase. Remember, there's little checkpoints at each of those. So we want to stop and actually cause apoptosis to occur if there's a mutation or something went wrong with the actual growth or the DNA replication at that point. If those checkpoints fail and something's causing it to not be able to do its job, just like the tumor suppressor genes, well, what happens is there's actually those mutated cells that have nothing's controlling it from dividing at a certain rate or at a certain speed. Also, remember, telomerase is actually allowing the cancer cells to continue to divide when they shouldn't. So that's what causes uncontrollable cell growth. They divide and divide so rapid and so fast. That's what causes cancer. So it's neoplasm is an accumulation of abnormally dividing cells. It actually forms a lump in the body called a tumor. Okay, so right here, this is called the ABCD rule. If you're checking for, let's say, for example, you have some sort of irregularity on your skin, you can actually do the ABCD rule and see if it may be cancerous. Number one, is it asymmetrical? Is that actual spot, whatever you're looking at, is it asymmetrical? Is it a perfect round shape, or is it completely unasymmetrical, which would be this guy right here? This is not a perfect round shape. It's actually very irregular. Number two, the border irregularity. So A, B, so the border. If it's not a perfect round shape and it's kind of got a lot of different shapes going on around it, there's a lot of irregularity to it. Okay, that's another red flag. Number three is C, ABC, the color. If it's dark, 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 and it's not one single color, it's something to be worried about. As you guys can see here, this neoplasm is very, very dark in these areas, but then lighter in this area, and it's very, very irregular. And last but not least is the diameter. If the new spot that formed is actually greater than 1/4 of an inch diameter, you want to get checked. So definitely, if you can put a quarter to it, and it's the size of a quarter or larger, definitely go get it checked. Okay, so proto-oncogenes. These are basically genes that can have or where tumor-causing mutations can occur. That's what they're called, proto-oncogenes. But an oncogene is actually a proto-oncogene that has that tumor-causing mutation. It actually already has that mutation occur on it. So, when is mitosis dangerous? Okay, because remember, we're talking about neoplasms. So if there's a mutation and those checkpoints aren't working, it becomes dangerous. Okay, because we don't want mitosis to keep happening if there's a mutated cell. So malignant neoplasms are progressively worse and can metastasize. What metastasize means is that it can travel. These are actually going to be very, very dangerous ones. So benign ones usually do not travel and they actually grow very slowly. So these are not as dangerous, but a malignant tumor is very dangerous because, like I said, number one, it's going to grow very, very quickly and uncontrollably. And number two, it's able to move throughout the body. So let's say I have a malignant tumor that starts off in my arm right here, near my armpit, near my lymphatic system, my lymphatic nodes. And then if it gets into my lymphatic system, then guess what? It can go throughout anywhere in my body because my lymphatic system runs through my head to toe. So those are a lot more dangerous. Populations are extremely high here as far as cell goes, and it has an altered cytoplasm and plasma membrane. That's why, once again, it is not going to stay anchored in place. That plasma membrane is going to allow the actual cell to actually rupture and open up, and once again allows it to actually go in and out of the circulatory and lymphatic system. So this is benign versus malignant side by side. Once again, benign neoplasms grow slow and stay anchored. So if this is actually a benign tumor, it's going to stay here and it's going to grow very, very slowly. Malignant, though, right here, they grow very, very fast and they can break away, as you guys can see right here. And once it breaks away, in this example, it can actually bleed into, this is actually the cardiovascular system where all the red blood cells are. And if it gets through here, it can attach anywhere else and cause a tumor anywhere else in the body, but also it can actually cause blood cancer as well. Very, very dangerous to actually have those. And binary fission. So it's cell division in the prokaryotic cells, because this whole time we've been talking about eukaryotic cells, which are plant and animal cells. Now let's look how binary fission occurs in prokaryotic cells. These are how prokaryotes actually divide. It's asexual reproduction, basically meaning you only have to have one cell. You don't have to have a male and female. It's just that one cell. And they actually split completely into half into two new cells. So here's our prokaryotic chromosome, is the plasma membrane, and then we actually have a cell wall protecting that actual prokaryote. And then duplication of chromosomes, it still duplicates, but then actually all of a sudden, then we actually have division into two new cells. That's actually the simplest of all divisions, and that's called binary division. So at this point, we've learned the difference between chromosomes, we've learned about genes and different traits, and we're able to describe the three stages of interphase, G1, S, and G2. We described the different behaviors of chromosomes during mitosis, and we talked about uncontrolled cell growth, which can lead to cancer and tumors. And we also did a brief section on binary fission in prokaryotes because it is the simplest of all divisions.